This lecture for microbiology is on Chapter 11, Prokaryote Diversity. The questions that we will answer in this chapter are, first of all, what is the scope of prokaryote diversity? Second, what are the key genera and characteristics of domain Archaea? And then third, what are the key phyla and representative genera of bacteria, and what are their essential characteristics? First, I want to start with a quiz question. This is something I haven't told you yet, but I want you to take a guess. And I'll mention right now that you should write down what you think is the correct answer while you're watching the presentation, and then after the lecture, go into Blackboard and complete the quiz, which I have provided a link to. Okay, so what is your guess? Write that down. Okay, well, the correct answer is thousands. There are thousands of bacterial species, in a typical gram of soil. A gram is not very much. It's about a cubic centimeter of soil. It seems like you couldn't pack that many kinds of bacteria in that small of a space, but they're really small and really diverse, and you can find thousands of species of bacteria in a gram of soil. That gives you a little bit of an idea of how diverse bacteria are. If 1 gram of soil can have thousands of species, just think how many species of bacteria there are throughout the world. So, in general, indeed, there is an astounding number of species of bacteria. Studies using PCR from habitats in the soil indicate thousands of species of bacteria per gram of soil, and not only that, but roughly 40 million cells of bacteria in that much soil. However, despite our knowledge of this diversity, it's still true that many bacteria have not been identified or characterized because they haven't been cultured. They haven't been able to grow them in culture. And I want you to think for a minute about why that would be. Secondly, think about, well, how do we know that there are this many species if we can't grow them in culture? Well, first of all, I mentioned here that PCR studies are being used to determine the number of species of bacteria. Molecular biology, including PCR, has created a revolution in our ability to study microbial communities like fungi and bacteria in habitats like soil. We can use PCR and amplify all the different bacterial DNAs in a soil, but all that tells us is that there are thousands of different bacteria. It doesn't tell us exactly who they are and what they're doing or what they look like. We know that there are different DNA sequences, and we can actually come pretty close to telling what kind of bacteria they are related to, but it doesn't tell us anything about their characteristics and their habits in the soil or what they look like. Why can't we culture all of these bacteria? Well, there are several potential reasons for that. One is that some bacteria may need special nutrients that we are not aware of. If we're not aware of it, we can't put it into a culture medium. Secondly, many bacteria may be part of complex food chains requiring the products of other bacteria. So, for example, bacterial species A has a waste product that is consumed by bacterial species B. Meanwhile, B makes a waste product that is consumed by species C, and species C produces a waste product that might be consumed by A or by another bacterium. Therefore, all of these bacteria cannot grow separately. They need to grow together because one depends on the other and another depends on another. Culture techniques in laboratories usually require growing bacteria separately in separate colonies. One aspect of microbial diversity that I want to mention is variation in size. Bacteria are mostly very small, less than 10 microns, and usually with a diameter of about 1 micron. However, there is some significant variation in size. Recently, it's been discovered that there are some bacteria that are gigantic, at least from a bacterial perspective, up to 750 microns in diameter. This is getting to be visible to the naked eye. You can almost pinch one between your two fingers. Thiomargarita is the genus name for this group of bacteria, and they are gigantic. They're found in lakes, and they provide some mysteries for scientists, one of which is how they can get nutrients into the middle of their cell if they are so fat. Because they depend completely, as prokaryotes, they depend completely on diffusion to get nutrients in. So how do they get nutrients into the middle of their cells if they're so large? Well, one thing is that they pack all of their metabolism and cellular machinery, their organelles, around the exterior of that large cell, right near the surface of the cell, rather than having it in the middle. So then diffusion is sufficient to allow nutrients to reach those organelles and other cellular machinery from the outside of the cell. Okay, this slide is meant to remind you briefly that we have a three-domain system in bacterial and archaea and eukarya classification. Those three groups of organisms are divided into three domains: eukarya, archaea, and bacteria. What we're going to focus on in this lecture is the diversity and characteristics of the prokaryotic groups, bacteria and archaea. Now, we're going to depend heavily during this lecture and the next one on the taxonomic hierarchy, so you need to make sure to know it. The taxonomic hierarchy is shown here with some examples. Remember, the highest level of the taxonomic hierarchy is the domain. Next down below that is the kingdom, at least in the domain eukarya. Below kingdom level, we have phylum or division. Plural of phylum is phyla. Below phyla, we have classes. Below classes, we have orders. Within orders, we have families. Families have multiple genera, and genera is the plural word for genus. And then within each genus, we usually have more than one species. This is the taxonomic hierarchy. And to show you one example, the domestic dog is in domain eukarya, kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Canidae, genus Canis, and species is Canis familiaris. Remember, species names have two words. The first word is the genus, second word is the specific epithet. Those species names are always always italicized. The genus name is capitalized. The specific epithet is not. Okay. We're going to be focusing today on major phyla within domain bacteria. We're also going to touch on domain archaea, but mainly we're going to talk about major phyla in domain bacteria, and then representative genera of those phyla. We're mainly not going to talk about the intermediate levels of the hierarchy. So first, a few words about domain archaea. We've talked about them a bit already. I'm just going to add a little bit more detail today. One thing that is different about the archaea compared to bacteria is that they do not have peptidoglycan in their cell walls. Secondly, most of them are extremophiles. That's PHILES. That means they live in extreme environments. And beyond that, the archaea are really diverse in their shapes, in their staining reactions, in their reproduction and how they make more copies of themselves, and in their physiology. I want you to know about three groups. I've already mentioned these groups, but today I'm going to introduce the names of three genera, one each for each of these three groups. And you need to know the names of these genera and remember that they are archaea and that they belong and which group they belong to. The example that I want you to know of the hyperthermophiles is the genus Pyrodictium. And you can partly remember this one because pyro means fire, which should remind you of this genus's affiliation with heat. These archaea live in very hot water, for example, in hot springs associated with volcanic areas. Methanogens are a group of bacteria that are also associated with hot water. These are archaea that generate methane. That is why they're called methanogens. They generate methane, they produce methane as a byproduct of their metabolism, which takes place in hot water, usually at the bottom of the ocean, where there are hydrothermal vents, where ocean water is superheated in response to magma from the core of the earth pushing up to the surface. Methanobacterium. Now, don't be tricked here into thinking that this is a bacterium. Just because the name says bacterium, it is not a bacterium. It is an archaea. Similarly, halobacterium is not a bacterium. Halobacterium is an archaean that is a halophile, an extreme halophile. These are some of the archaea that live in very, very salty water. For example, in the salt lakes of the western United States. And you need to know all three of those genus names. Remember that they're archaea. Okay, now the rest of this lecture we're going to be focusing on the characteristics and representative genera of several key phyla from the domain bacteria. First of all, these two, phylum Firmicutes, which contains gram-positive bacteria that have low amounts of G and C in their DNA relative to A and T. The amount of G and C versus A and T is something that has been shown to vary among different kinds of bacteria. It's easy to figure out. And it turns out it has a strong affiliation with certain groups of bacteria. The second gram-positive group of bacteria we're going to look at is the actinobacteria. They are gram-positive, but they have high GC content in their DNA. A third group is the proteobacteria, phylum Proteobacteria. Fourth, we're going to look at phylum Cyanobacteria. These are the photosynthetic bacteria, the oxygenic photosynthesizers. Oxygenic means producing oxygen, as normal photosynthesis does. And then fifth, we're going to study the phylum Chlamydiae. From each of these phyla, you need to know a few general characteristics, the names of particular genera, and then some characteristics of those genera themselves. Okay, here's another quiz question for you to write an answer down to. Oxygenic photosynthesis occurs in which bacterial phylum? We just talked about it on the previous slide. Is it Cyanobacteria, Firmicutes, Proteobacteria, or Actinobacteria? And I want to mention that because you have access to this presentation and you can go back and look at the answers in the presentation and listen to me, you need to get every quiz question correct on Blackboard in order to get credit for this lecture quiz. Okay, what is the correct answer here? It is cyanobacteria. They are the photosynthetic bacteria. And which phylum contains the low GC gram-positives? Is it the cyanobacteria, the Firmicutes, Proteobacteria, or Actinobacteria? I'll give you a few seconds to write down an answer. Okay, now remember, this one is from just a couple of slides back. The low GC gram-positives are the Firmicutes. So this answer is Firmicutes, number 2. Okay, a little bit more information about the Firmicutes. First of all, a reminder, these are a couple of things I already said. They have low GC content in their DNA, they are gram-positive, and then something I hadn't mentioned yet is that they are all chemoheterotrophs. And you should know from the metabolism lecture what we mean by that. Secondly, I want to point out the name of this phylum, Firmicutes. Firm means tough, and cute refers to not good looking or attractive, but rather cute, like cuticle. It refers to the skin. They don't have skin, of course, but they do have an external structure sometimes that is very tough. In fact, these are the bacteria that form endospores. Not all firmicutes make endospores, but a lot of them do, including a couple of the ones mentioned below. The order Clostridiales, on this, as you can see on this diagram, and the order Bacilliales, or Bacillales. This includes the Staphylococcus bacteria and Clostridium. The key genera that I want you to know the names of are Clostridium, Bacillus, that's spelled B-A-C-I-L-L-U-S, Staphylococcus, Streptococcus, and Lactobacillus. These are the 5 genera. You need to know that they belong to this phylum, that they have these characteristics listed here, that some of them form endospores, especially Clostridium, Bacillus, and Staphylococcus. And then we're going to talk here in the next few slides about unique characteristics of each of these phyla. Each of these genera. Okay, first a couple of notes about Clostridium. First of all, Clostridium is indeed one of the genera that it produces endospores. We talked about endospores before. Endospores are tough external coatings on cells that allow survival of bacteria through very difficult conditions. Clostridium is one of those bacteria that does this. These guys are obligate anaerobes. You should know from previous lectures what that means. It means that they can live in the absence of oxygen and only in the absence of oxygen. They cannot tolerate oxygen. So they tend to live in places in soil where there's not very much oxygen. They can live in your large intestine sometimes, and they can live in deep wounds, not in surface wounds where there's a lot of oxygen, but deep in puncture wounds. They are indeed common in soils. That's one of their most common habitats. And the example that I want you to know about especially is Clostridium botulinum. Clostridium botulinum is a species that has a big impact on humans because it can cause the disease called botulism. And we will cover botulism in a little bit more detail after the next midterm exam. But for the reason that it causes this disease, it is the main target of sterilization in commercial canning operations, which I've mentioned before. The way that disease botulism works is that this bacterium produces a neurotoxin that freezes your nerves. And it is this toxin, in fact, that is used in Botox procedures. You probably have all heard about Botox procedures. The way this works is that a doctor will talk to someone about the wrinkles on their face, for example, and you might complain that you have some wrinkles near your eyes that you want taken care of, and a doctor will take some botulinum toxin from this bacterium and inject it into your face. And what this does is it kills the nerve, deadens the nerve associated with the wrinkles. Wrinkles are sometimes caused by muscles that are pulling, and so if you kill the nerves, you can relax those muscles and get rid of the wrinkles. Now, if you didn't already think this was a bad idea, let me tell you, it's a bad idea to do this to your face. Please don't ever inject botulism toxin into your face. Why, you might ask? Well, one reason is that it's a bacterial poisonous toxin. That's just not something you should inject into your face. Occasionally there are accidents that happen where the toxin gets out into your body and you can be poisoned by that. Also, when you get older, your wrinkles will be beautiful. You don't need to get rid of them. Okay, a second member of this phylum you need to know is the phylum Bacillus. The genus Bacillus. This genus also makes endospores, And the cells of this bacterium are rod-shaped, so these are endospore-producing rods. The image on the right shows one of these bacteria as it's hatching out of an endospore, presumably after being rehydrated. These are also common in soil. Very few of them are pathogens. In other words, they don't cause a disease. You should know the word pathogen. I'm going to use it a lot in the next couple of weeks. Pathogen is an organism that causes disease. Very few bacillus cause diseases. The most famous example of bacillus is Bt, which is Bacillus thuringiensis. We talked about this bacterium just a couple of days ago. This is a bacterium that makes a toxin that kills insects, and it's this toxin that is produced by a gene that we have learned how to put into plants. If we put this into a cotton plant or a soybean plant, then that plant can produce a toxin that kills insects. This has some benefits, but it also has some downsides because it kills beneficial insects and it causes insects to evolve resistance to this chemical. Okay, third up in the phylum Firmicutes is the genus Lactobacillus. Lactobacillus is really important for human health. I've mentioned this before a couple of times. Many species in this genus are important members of the normal microbiota of the human body, especially the colon in males and females, and then the vaginal area in females. These bacteria produce lactic acid as a product of fermentation, and that is how they protect us. The lactic acid that they produce prevents other bacteria from growing, and it helps protect you from disease-causing bacteria and disease-causing yeast. The acidic environment around them inhibits other microbes in the human body. In addition, we use these bacteria to make food. The lactic acid from their fermentation adds a tangy or sour flavor to many fermented foods such as sauerkraut, yogurt, pickles, and other foods. Okay, Staphylococcus is also in the phylum Firmicutes. This is a genus that you need to know. You may recall that the name of this genus means cocci in clusters. It means round cells, that's the coccus part. And staphylo, or staph, means clustered or clumped. The picture here shows what I mean by that. Many of these bacteria are pathogens. They're also good at growing in difficult conditions, and by difficult I mean drier than average conditions, saltier than average, and one example of one place where they can grow is on your skin. Staphylococcus bacteria are somewhat common on your skin. And then finally, Streptococcus. Streptococcus are cocci, round cells, as well, but they grow in chains or strips, which is also indicated by their name. These are mostly aerotolerant anaerobes, so they are able to grow in a wide range of conditions. They don't thrive under oxygen, but they also can survive it. These bacteria cause lots of diseases, even more than the Staphylococcus. Strep in general, Streptococcus or strep bacteria cause a whole bunch of diseases. Okay, the second phylum you need to learn is the actinobacteria. These are also gram-positive, like the Firmicutes, but they have high GC content in their cells, in their DNA. The actinobacteria has two genera that I want you to know, Streptomyces and Mycobacterium. One thing I want you to notice right away is that these two names both have MYC in their name. And that's one way you can help try to remember that they belong to the actinobacteria and that they belong together. What does MYC mean? Think about that for a minute. Well, one thing you may have thought about is that MYC often means fungus. It refers to mycology. And in fact, Streptomyces does kind of look like a fungus when it's growing. It grows in little threads and it makes spore-like structures. Both of those are fungal traits. So Streptomyces does kind of grow like a fungus. Mycobacterium, however, has a different reason for being called myco, and we'll get to that in a minute. So Streptomyces, it is filamentous, meaning that it grows in a branching fungal-like filament. You can see a picture of that to the right, almost like a little ribbon. It also makes conidiospores, which are round spore-like cells that can fly through the air. Most bacteria cannot disperse through the air, but Streptomyces can, which makes them somewhat like a fungus. They make spores. They are common in soil, just like Bacillus and Clostridium. And then finally, what is important to note about them is that this particular genus has produced most of our useful antibiotics. Remember, antibiotics are antimicrobial chemicals that we harvest directly from natural sources, natural species. Well, we haven't had a huge success doing that from a lot of different kinds of species, but Streptomyces has given us a lot of success. The second genus that we need to know in this phylum is Mycobacterium. Mycobacterium have a unique resistant waxy cell wall, which makes them very difficult to kill. And this is why they're called myco. In this case, myco means waxy. It refers to that waxy substance that is deposited in the cell wall. Because they are difficult to kill, sometimes they can cause nasty diseases, including tuberculosis and leprosy. We'll talk about both of those diseases in more detail after the next midterm exam. Okay, here's a quick quiz question for you. What is the prefix that means ray, R-A-Y, and refers to the bacterial phylum that includes species that form star-shaped colonies that are often fungus-like? Well, I haven't actually said this yet, but the prefix here is actino, A-C-T-I-N-O. This is the prefix that begins the name of the actinobacteria, which is the group we were just reviewing. So there's another way that you can remember the name of this phylum, and the fact that both of the genera have mic in their name is that these bacteria form star-shaped colonies that are kind of fungus-like, and that's what actino means. Okay, a third group you need to know is the phylum Proteobacteria. Proteobacteria are named for the mythical Greek god Proteus, who could assume many shapes. The reason for that is that the phylum Proteobacteria is very diverse. It's the largest phylum with the most types of bacteria in it. Most of these are gram-negative chemoheterotrophs. And the key genera that you need to know are Rhizobium, Spirillum, Pseudomonas, Escherichia, and Salmonella. You should be recognizing a lot of these names. They've come up quite a bit in the class already, in some cases, and now we're going to put a little bit more information on them. First of all, Rhizobium. This is one that I don't think we've talked about so far. These bacteria have a unique characteristic, which is that they live in what are called nodules on the roots of plants. And you can see a picture of this to the right. This is the root of a plant, and those little balls clinging to the roots are nodules. Nodules are bacterial houses that are created due to a partnership between the plant and the bacterium. The bacterium interacts with the root and signals, and then the plant forms a fleshy structure that the bacteria can live inside. They feed the bacteria with sugars from their roots that they make in photosynthesis and then bring down. In return, the bacteria provide a really unique service for the plant, and that is that they fix nitrogen and give some of that fixed nitrogen to the plant. Fixing nitrogen means you take nitrogen out of the air in the form of nitrogen gas and turn it into a mineral form that can be given to a plant. In this case, the form is ammonia, or NH3. What does rhizo mean? The letter is 4-letter word. So the answer is a 4-letter word, and it actually is found on this slide. So look around. Do you see any 4-letter words? Rhizo means root. So Rhizobium is a root-loving bacterium. Okay, also in the phylum Proteobacteria, I want to mention Spirillum. Spirillum is a genus that is chemoheterotrophic, and unique among the bacteria that I've mentioned so far, it has a spiral cell shape. The spiral shape is not something we've come across very much, so I wanted to throw this one in there. And it's easy to remember this name, Spirillum, because it has a spiral cell shape. These cells also have flagella on both ends, which allow mobility in both directions. And usually they live in stagnant, organic-rich water, like a puddle that has dead leaves in it. There is where you'll find Spirillum. Also in the Proteobacteria is the genus Pseudomonas. Pseudomonas is a really diverse group of bacteria. It's abundant. It has lots of different characteristics depending on which species you're talking about, and it lives in lots of different kinds of habitats. However, many of them are opportunistic pathogens, meaning that they're not always pathogens, but when there's an opportunity presented, they may become pathogenic. So for example, if there is a weakened patient in a hospital, for example, a burn patient who has come in whose skin is severely burned, Pseudomonas might cause an infection in them. Someone with a compromised immune system for another reason. Maybe they have an immunosuppressive disease like AIDS. Maybe they're taking drugs to suppress their immune system for an organ transplant. Opportunistic pathogens attack people like that. And I'm going to come back to that idea over and over again. So right now, take the time to remember, what do I mean when I say opportunistic pathogen? These bacteria also have polar flagella, flagella on one end. Okay, and then finally in this phylum are the genera Escherichia and Salmonella. They share several things in common. Both of them are enterics, which means that they live inside your digestive system, your large intestine mainly. Both of them are facultative anaerobes, which means that they can live without oxygen, but they don't have to. And then gram-negative rods characterizes their cell shape and staining reaction. They're bacilli, meaning they're rod-shaped, and then they're gram-negative, so they would have a gram-negative reaction to the gram staining. Escherichia is our key example of the genus E. coli is our key example here of the genus Escherichia. These bacteria are mainly important members of your normal microbiota living in your large intestine. You may have heard of E. coli because it gets in the news once in a while due to an E. coli outbreak where it causes poisoning and sometimes even death. However, most of the time it's harmless and even very helpful to us. It's just that once in a while a pathogenic strain is found in beef or other foods that makes us very ill. Salmonella, on the other hand, is often pathogenic. Usually when we find it around humans, it's causing some sort of gastrointestinal disease. And we will talk a little bit more about both of those organisms as disease causers after the next midterm exam. All right, 2 more phyla. One of them is cyanobacteria. These are named because of their color. These have blue-green colored cells. They used to be called the blue-green algae. They're not algae, so someone changed that name. Cyan is a color. You may remember having a cyan crayon when you were a kid. All of these bacteria perform oxygenic photosynthesis, where they produce oxygen during photosynthesis. Many of them also fix nitrogen. Remember what this means? We learned about it with the Rhizobia bacteria. This means that they can take gaseous nitrogen out of the air into gas and turn it into a mineral form, ammonia, that can be used by an organism. These are also gram-negative. All of the groups, the first 2 groups were gram-positive. These last ones are gram-negative. Anabina is the only genus name I want you to know. There are lots of genera of cyanobacteria. I just wanted you to know at least one. Anabina, Anabaena, looks like this. You can click on the link yourself to look at the video of Anabina swimming. One thing I want to point out is the cells that are called heterocysts. You notice how some of these bacterial cells are brighter and rounder than others? Well, those round, bright ones, those are the site of nitrogen fixation. Heterocyst means different cell, and in fact, these cells have a different function. Notice also that Anabina is colonial. Multiple cells are strung together, and they, to some degree, work together to help each other. Okay, quick quiz question. What is the prefix that means blue-green and refers to the oxygenic photosynthesizing bacteria? Give you a minute to write that down. Okay, good. That's cyan, C-Y-A-N. All right, and then finally we're going to cover the phylum Chlamydiae. Chlamydiae is unique among bacteria in having very little or no peptidoglycan. But it does have the genes to produce it, it just isn't doing it. This is of interest to scientists to figure out how these genes are present but not expressed. These are also gram-negative bacteria, but they form cocci. And the key genus is Chlamydia, C-H-L-A-M-Y-D-I-A. The main characteristic of chlamydia that you need to know is that multiple species of chlamydia cause disease, and multiple kinds of disease. Trachoma is a blindness disease caused by a chlamydia species. There are multiple different sexually transmitted diseases caused by chlamydia. There's a form of pneumonia, and then several other diseases. I put these funny pictures on here about chlamydia, public service announcements. Sexually transmitted diseases caused by chlamydia are at an all-time high, so some local and regional governments are trying to get out ahead of this and prevent this problem. So Canada here, the Saskatchewan region put up this public service sign that proclaimed them the chlamydia capital of Canada, which they were at the time. On the right here is a public service poster from the state of Virginia. It says, "So you think chlamydia is a flower!" Exclamation point. And I'm not sure that's a very effective poster. Most people don't think chlamydia is a flower. Also, the poster doesn't really very easily tell you what chlamydia really is. So I don't know that that's the best poster. Okay, a few clicker questions to finish up. Which of these genera listed here is not a member of the actinobacteria? Now, I'm not going to tell you the answers to these last few questions. I want you to write down answers that you think are correct, and then you can go back and study the lecture to make sure you get it correct, and then go to Blackboard and complete the quiz to get credit. Okay. Which of these genera forms branching fungus-like filaments? I'm going to give you a hint on this one. It's the one that has some letters in the name that refer to fungus. Which of these genera is a source of many commercial antibiotics? This is another one with the words for fungus in the name, but that doesn't exactly narrow it down in this list here. Which genus has a very unique, highly resistant cell wall structure? Which of these bacteria, strangely, seems to lack peptidoglycan in its cell walls? Is it Clostridium, Chlamydia, Mycoplasma, or Rickettsia? Which phylum contains bacteria that are all photoautotrophs? Is that Proteobacteria, Cyanobacteria, Chlamydia, or Actinobacteria? Which of these genera is not a member of the domain Archaea? Now, you might think it's the one with the bacterium in the name, but no, it's not. The answer is one of the other three. Think about this one. Which of these bacteria, if you find it in water, might suggest fecal contamination of the water? In other words, poop is getting into the water due to poor sanitation. I'm going to tell you right now, there are two possible correct answers to this question. I would not use such a question on an exam. Which genus of the proteobacteria associates with the root nodules of plants? Now here you need to look for the Latin word for root among the names. Which genus forms resistant endospores and may cause human sickness? Clostridium, botulinum, chlamydia, and streptococcus. Now notice on this question it's asking for a genus name. That should be a helpful clue. Which genus is not a member of phylum Firmicutes? Clostridium, Bacillus, Pseudomonas, Streptococcus, or Staphylococcus? Okay, this is the end of this chapter. Please go to Blackboard and complete the quiz and make sure you get all of the quiz questions correct in order to get credit for the quiz.